wanted to welcome everyone to our second Informatics Grand Rounds of the fall here at Johns Hopkins University School of Medicine. I'm Paul Nagy, one of the Informatics faculty here at Hopkins and Deputy Director of our Johns Hopkins Technology Innovation Center. I work closely with the Precision Medicine Initiative and enjoy training our students and faculty alike on the Precision Medicine Analytics platform in our informatics education program. I'd like to recognize our excellent master's, PhD, and certificate students who are joining us today for our grand rounds and congratulate you on nearing the end of our first quarter. For others who are joining us from around the globe, welcome, and please feel free to send any questions our way via the question and answer tool. Dr. Davidson and myself will be reviewing the questions and offering an opportunity for some of you to pose them directly to Dr. Ray over the course of the next hour. This Grand Round series is offered for continuing medical education credit for those physicians eligible to receive CME. I am thrilled to be introducing our Informatics Grand Round speaker for today, Dr. Stuart Ray. He's the Vice Chair of Medicine for Data Integrity and Analytics and a professor in the Division of Infectious Diseases with secondary appointments in viral oncology and health science informatics. He's been at Johns Hopkins since 1990. When he graduated from the Vanderbilt University School of Medicine, Stu completed his internship and residency at Johns Hopkins Hospital, continued on as an assistant chief of service and fellow in infectious diseases. He has served as a preceptor for several of our graduate students in informatics, along with the many other hats he wears at Johns Hopkins Medicine. Dr. Ray researches the influence of viral evolution on viral pathogenesis, concentrating in particular on complex RNA viruses such as hepatitis C virus and human immunodeficiency virus. He has published approximately 100 scientific articles on these topics. And on a personal note, I've had the pleasure of working with Stu for many years, maybe not all the way back to 1990, but at least the last 10 years on clinical data analytics and the Johns Hopkins Medicine Data Trust. Recently, we've worked closely on Cadre, the COVID data research evaluation team, and JH Crown registries to accelerate biomedical discoveries on COVID during the pandemic. Along with other colleagues, I'm deeply impressed with his subject matter expertise in infectious disease, data analytics, basic science, and informatics, all of which have come to the fore now more than ever during COVID-19. He'll be speaking to us today about clinical metadata in the pandemic response. Stu, thanks for joining us today.
Well, thank you, and Ash and all who've helped organize this. It's a pleasure to speak to the group, and I'm feeling a little bit like I'm bringing coals to Newcastle when I'm talking about informatics to this group. And I'll go ahead and start sharing the slides and provide a couple of disclaimers. So in addition to my disclosures, which I should show, I'll say that my talk is going to be focused a bit on the work that I've been engaged with, because I think that's a perspective that I can provide, that's new to this crowd. I will be focusing very tightly on the informatics issues. What I'd like to do is lift up some issues around gathering viral genomics, and then the issues around gathering metadata, and some of the issues in terms of privacy and our ability to share the data, especially in a pandemic response. This group is coming all over the world. I'll say that the data that I'll talk about will be US-centric, and some of my perspectives will be US-centric, but I'm happy to discuss issues of broader significance. And my hope is to lift up some issues about how we can understand the intersection of the viral genomics with clinical metadata as we try to understand and intercede on the impact of SARS-CoV-2 on the human population. So I'll be talking about this intersection of viral genomics with the metadata, and hopefully this will stimulate some thoughts about this. So getting this back into context, there we go. The SARS-CoV-2 epidemic, COVID-19, has provided some surprises, and I think it's worth noting these, not that I'm going to explain them, but that we still have some unanswered questions. So we know that there's pre-symptomatic transmission, and this is a major distinguishing factor between this virus and the one that caused the epidemic in 2003. It's also becoming clear that droplet and in some situations aerosol transmission dominates transmission here, that masks work and they're not perfect, but what is, that there may be some fomite transmission, but its role is less prominent, at least it seems, than we've thought with flu. And the droplet plus precautions, where we use both contact and mask precautions with face shields in certain situations, seems to do a great job of preventing nosocomial transmission. We have systems that encourage ad hoc off-label use. And so one of the hallmarks of this epidemic, unfortunately, has been the ad hoc use of treatments with poor evidence. And it's too hard to do randomized clinical trials. And I think we're realizing that and thinking that as we move forward, we need to find a way to gather robust data in the context of interventions for a pandemic like this, so that we'll know how to move forward with an evidence base. And then, of course, the publication pace and the increasing role of preprints, and that will be highlighted in some of the things that I talk about. Another context from which I'll be speaking will be as a member of this group. So there's a committee that met with the National Academies of Medicine specifically to talk about modernizing pandemic response strategies. And the title of the report is Genomic Epidemiology Data Infrastructure Needs for SARS-CoV-2. You can see the links here in the bottom right-hand corner. This report is now available. The PDF is free. And I'll be borrowing a couple things from it, but also want to say that we spent some time in this report going into detail about the issues around sharing metadata and the need for a more robust infrastructure for this. It included Diane Griffin as the chair and Lisa Brown as the study director, and so is a work of a lot of people, and we moved pretty quickly. This table summarizes some of the considerations for questions that we'd like to address using genomics and metadata. Things like, what is the structure genetically of the outbreak? Is it multiple introductions? Where's the virus coming from? Is it due to local spread or further introductions? Is there super spreading, and how would we recognize that? Is the virus changing over time? Is it evolving? Is resistance becoming an issue, both to drugs and vaccines? And then are there strains that differ in their pathogenesis, response to treatments? You can imagine the issues that come up. And so this summarizes some of the things to think about, both in terms of what the viral genomic sequence data might need to be, and then what the linked metadata needs would be to address these sorts of questions. I think we also have to realize that often, as will be the case for the data that I discuss from Johns Hopkins, often we're dealing with a convenience sample, a sequencing of specimens that are available rather than a true population-based sample. And this is one of the challenges we have, at least in our setting, in trying to do this. Now, in order to talk about this virus, I think it's worth talking about its origins. And so on this phylogenetic tree, where vertical distance means nothing, horizontal distance on the tree is genetic distance. You can see the isolates from the epidemic of SARS-CoV 20 years ago, and then you can see some bat coronaviruses. Here's the novel coronavirus with strains from late 2019 and early 2020 from Southeast Asia. And then you can see other bat coronaviruses. You can see MERS-CoV, and then some of the respiratory coronaviruses, HKU1, OC43, that cause about a third of colds in humans. And so these viruses all provide a context for the source of this virus. And I'm going to touch for a moment on some of the virologic characteristics and then its origins. So this is a beta coronavirus. There are 4 major lineages of coronaviruses, as you see listed here. And this is a lineage B sarbecovirus. Its genome has 30,000 nucleotides. This will come up a few times in this talk. There are multiple open reading frames and about 30 potential gene products, cleavages of the non-structural proteins and structural proteins. There are accessory proteins which interfere with the human immune response, for instance, interferon signaling. The spike protein will be a major component of this discussion, but as depicted here, there's about 30,000 nucleotides in this genome. So the structure of the virus, I'll just highlight that the spike protein sticks out from the surface of the virion. The virion is about 120 nanometers across, so about 1/20th the size of a red blood cell. And the spike protein sticks out prominently, giving the virus its name, the coronavirus, or looks like a crown on electron microscopy. And so this spike protein will be a part of our discussion, but there are multiple proteins in each virion. Now, as a somewhat narcissistic sidelight here is that when I was a fellow transitioning to faculty in the late 90s, I wrote a piece of software called Simplot, and it's been used in about 2,500 publications to date. And it generates these plots that show similarity of one genome to another as a sliding window. And so in this first report of this virus genome, they compared the SARS-CoV-2 genome to other existing genomes. And so what they found was that whereas the SARS-CoV genome from 2003, this red line that dips low here, the bat coronavirus RATG13, which had been isolated in 2013 from feces in a cave populated by bats in Yunnan province, about 1,000 miles from Wuhan, that this virus was the most similar to SARS-CoV-2. Even though it's most similar, and it's about 95% similar on average across the genome, with a genome size of 30,000, that is thousands of differences between this virus and SARS-CoV-2. And so it was clear that this was not an immediate zoonosis from this virus to humans, both because the virus is so different and also it was found 1,000 miles away. We really don't have the proximate ancestor of the SARS-CoV-2. We'll talk a little bit more about other ways of thinking about this. And again, I'm describing this because when people hear about genomic data being compared and linked with metadata, it's important to consider how the data are acquired and how we analyze it. This is another illustration from a different paper from The Lancet, also from late January, early February, when these genomes were first being sequenced, showing the similarity, but also noting the deep dips in similarity between this new virus and these existing ones, with the spike protein being the most divergent across all isolates, which is not a big surprise. Often viral envelope proteins differ a lot from isolate to isolate. And you can see that these dips go below the 50% threshold, which means that less than half of the amino acids in each of these sliding windows is identical between that isolate and SARS-CoV-2. And another, and I'm showing these as a sequence, not because we want to focus on these details, but I'll show in the next one why this is important, because when people sequenced coronaviruses from pangolins, what they found was an area where the SARS-CoV-2 sequence was more similar to an isolate from pangolins than from the bat coronaviruses, raising the question of whether pangolins might have been a intermediate host, or perhaps a coronavirus from pangolins might have contributed a segment of the genome. This is important for a couple of reasons. One is that it suggests that mammals can be infected with more than one coronavirus at a time for the recombination to have occurred. But also for us to understand where this virus came from. I would say that this is not a settled issue, and the consensus among virologists is it's not clear that we have close enough ancestors to know where this virus came from, but it does look like it evolved naturally in mammal hosts. Finally, this figure illustrates some of these issues in detail, where these brackets correspond to the trees on the right side. And I'll just summarize the paper by saying that this shows anomalous clustering in different parts of the genome, suggesting that there has been recombination in the lineage of coronaviruses, at least in the past. And a recent paper, a very nice paper that involved Eddie Holmes and others, suggests that the most recent common ancestor for this virus, SARS-CoV-2, is probably at least decades, if not hundreds of years old. That it's not a very recent recombination event based on the data that we have available. So This is what it's all about. This is the genome of SARS-CoV-2, obviously not something that humans read well, but it's just letters A, C, G, and T. And if you made an RNA from this and injected it into a cell, there's a good chance you could get replication. So this sequence is sufficient for the entire program of SARS-CoV-2, which is kind of awesome, that it's like a little program script that will run a cell to make more of this virus if this sequence is available. So when we think about this problem of intersecting the viral genomics with the metadata, we want to answer some questions, and I've already touched on these with the major table from that report from the National Academies, but we want to understand this epidemic with the viral sequence in mind. And so it's important to understand how we generate those data. And so this, I'm going to summarize, is a project that we had here at Hopkins, where people from the Applied Physics Lab at Johns Hopkins, so people down near DC, people in the School of Medicine like Heba Mustafa, who runs our microbiology lab and stood up the SARS-CoV-2 genetic viral testing for us very quickly in March. Winston Timp, who's a leader on the Homewood campus in biomedical engineering in sequencing, next generation sequencing, and Michael Schatz, who develops software along with Steven Salzberg, one of the most highly cited computational biologists in the world, is on our faculty. And all these people, along with people in the School of Public Health like Justin Lessler and Shirley Roll, with support from the Vice Deans for Research in the Homewood campus, provided the expertise to do the work that I'll summarize. And so here's Heba and Tom Mihok and Peter Thielen working in March on getting genomics up. They used an Oxford Nanopore device this size that costs a few $100s, and they started doing sequencing. And initially, our costs for a genome were about $100 for a sample, and it was about 36 hours from sample to result using pools of PCR amplicons. I'd be happy to go into some detail about how this was done, but I'll just say that it's amazing that these days, when this was so hard a decade or two ago, we can now do a 30 kilobase genome in less than 2 days for less than $100. And so we started sampling sequences from here in Baltimore.
Dr. Ray, a question
Yes.
came through regarding this, and the question was, this is a lot of prolific research to have been done in a short period of time. Can you please speak to how some of the restrictions with lab research and on-site work impacted your team?
Sure. So it was a challenge to do this work given our concerns about transmission. Of course, this picture was taken before some of the precautions started getting taken. So this was very early this was probably late February, early March when this was just being stood up. As soon as the restrictions came in, of course, distancing and masking and other precautions were taken as throughout the microbiology labs. And we had to limit the activities in the lab to essential functions. The sequencing only required one person to be working on pipetting and that sort of thing. There's not a lot of multi-person work involved in this. And so we were able to segregate the functions well enough to maintain distancing and masking. But it was certainly a challenge to get some of the supplies and other things going. And we started out with the nanopore simply because we had it available, and ramping up some other technologies that I'll talk about later would have been harder. And so we were able to do a pretty self-contained team with existing resources as described here. So as we move forward then, what we would get, and these are just coded specimens. So this is Maryland, and this is Hopkins pathology, and then an ID number. And so these are just study IDs that we assign to specimens. So each row is one specimen, and the blueness indicates the number of reads at a given amplicon, and there are about 100 amplicons spanning the genome. And so what you can see is that for some of the specimens, like number 6, 7, 10, 15, 22, 31, 32, 33, these are all dark blue, meaning we had a high number of reads in every amplicon. Others, an exemplar being 17, we had very low number of read counts, too low to read this with confidence. So we would get a sequence, but not a sequence in which we have great confidence. This is depicted here graphically in these green bars, where higher bars mean better coverage. And so you can see that with this one, we got nearly 10,000 reads in every amplicon across the genome, whereas in this one, 17, we had less than 10 reads for most of the parts of the genome. So this would be a fail for number 17, whereas 6 was a success. Of course, once we have all these reads, we then have to resolve those into a sequence. And so a pipeline had to be used, and we leveraged, so the PCR part of it, the wet lab parts came from the Arctic consortium, but the software pipeline involved Arctic and then some protocols from NextStrain and GISAID. And so there are collaborations worldwide that have made a lot of this possible. And so we were able to leverage some of the software from existing protocols. We had to write a custom software pipeline to do some of this joining. And so you can see that whether we're using nanopore sequencing or Illumina, we had data coming into this pipeline. The pipeline was hosted on the Sci server, the astronomy server at Homewood, which has previously housed the database for the Hubble Space Telescope and other major astronomy functions, led by Alex Saleh and others. And so this data pipeline was used to clean up the data, normalize it, do variant calling, and then we had manual initially, and slowly we trained the system to do automated resolution of mismatches in the sequences, because there are certain patterns that became evident that we were able to automate in software. And so the variants were then identified, and then there was post-filtering, cleanup, and then phylogenetic clade assignments. We also did phylogenetics, which we can talk about some if you wish, and I'll try to demonstrate some of that for you. And ultimately, we deposited sequences in SRA, we deposited sequences in GenBank, and we deposited sequences in GISAID, the international database housed in Germany that's used to house influenza genomes, but has been repurposed to house coronavirus sequences from around the world. And of course, without having a centralized database, we really can't contextualize the evolution of this virus as it moves around the globe. And so this was a first point at which we were trying to assemble some metadata. And one of the stickiest points was, what do we do with dates? The date on which a specimen was acquired, and the implications of that for privacy and usefulness for science, and trying to balance those considerations when we're trying to deal with a pandemic in real time. So the output of all this was that we got genomes, and on the left here is a simplistic clade assignment. So these clades, there's a clade that arose in 2019, which was called 19, and there are two parts of it, A and B, and I'll show you these pictorially in a moment. And then there were some later sequences that arose in 2020. That clade is called 20, and it has multiple letters too. And so here are 4 genomes from our set. The red triangles indicate amino acid replacements in proteins, and the gray triangles indicate nucleotide changes that did not change the protein. This label is wrong. This should say synonymous gray and non-synonymous red. But you can see that there are some changes relative to the Wuhan strain, but overall, not a huge number of changes. And for instance, if we were looking at hepatitis C or HIV, there would be far more changes. And we'll talk about evolutionary rate here. But this virus is not changing very fast. So if we depict the first 100 or so sequences that we obtained here at Hopkins in the Maryland, DC area, and then put these on the tree globally. So this is a sample of 6,500 genomes from the 85,000 that currently are full length and high quality in GISAID. You can see that sequences in the Maryland, DC area actually spanned sequences from the 2019 clade that arose in Southeast Asia and populated some of the epidemics in the Pacific Northwest, and then these sequences from Europe, and then the red sequences from the US. You can see the color code here has indicated that it goes from China in purple through the earthy tones for Europe, and then the red for North America. And so our sequences here in the Maryland, DC area actually were drawn from epidemics around the world. And so we had a very diverse population of sequences, even in the first 100 that we sequenced here in Baltimore.
Dr. Ray,
Yes.
an excellent question came through. Can you please speak to the data sources that you're using to compare the Hopkins specific DC, Maryland samples to the ones internationally? How are you able to generate this visualization?
I will show you the visualization interactively in a moment. It's an important thing that these data, GISAID, you can actually sign up and begin downloading data if you'd like. And I'll show you the links for doing that. And then the GISAID data is being consumed into a tool called NextStrain that Trevor Bedford and his team on the West Coast developed. And so that tool is also free and also interactive. And I'll show you how to reach that, and I'll actually show you how this is done interactively. In order to take our data, which were not yet polished in April and May, we actually were able to download the NextStrain software, which you can do, and create a local instance where we were able to put pre-submission data into the model and visualize our own data. But you can actually do the same thing now. If you were to just click on some options that I'll show you in NextStrain, you could make the same graph right now. So I'll show you how this works. Now this is a rectangular cladogram, and what it shows you, again, horizontal distance is meaningful for genetics. The horizontal position is actually based on time, so each point is placed on its collection date, or in the case of our data, dates that have been randomly shifted by an amount that was allowable for this purpose. And then the colors indicate, as I said, country. And then you can see at the branches, there are indications of amino acid replacements that have occurred. So here, this clade is established by a variant in ORF1A, so one of the proteins that we've talked about, where there's a threonine to isoleucine change at position 265. And so there's a famous one here, ORF1B, spike. The spike D614G mutation is right here. It's overlain with some other things, but I'll show you. But this tool, the NextStrain tool, will show you what amino acids changes occur at each branch. So you can explore this. You can click, it's interactive, so you can click on these points and explore them more interactively. The same tree drawn as a radial cladogram shows you the distribution and how our sequences were sampled across the whole tree. And so this at least offered the possibility that because these variants differ genetically, we might be able to see some correlation with severity. And one of our questions was, did our patients who ended up on ventilators, who ended up requiring oxygen, who ended up in the ICU and some dying, was that associated with a particular genetic signature in the virus, or was that dependent on host factors or other characteristics of their infection? So I'll come to that in a moment. This is a plot of genetic distance on the vertical axis against time on the horizontal axis. This gives us an estimate of the evolution rate of the virus. And again, I'll show you how to do this interactively. But this plot, which is generated by the NextStrain tool, gives you a rate estimate, this black line, this regression line, of about 23, 24 substitutions per year in this virus. So this is quite slow. With SARS-CoV-2 changing at about 8 times 10 to the minus 4 substitutions per site per year. So this is this number divided by 30,000. For instance, H3N2 influenza A changes at about 5 times faster rate. So this virus changes at a slower rate. And I'll say parenthetically that if you wonder about what the error rate of a viral polymerase is, it's generally 1 over the length of the genome. And actually that same rule holds for almost all organisms. So the human polymerase error rate is about 1 over the length of the human genome. And that same equation is true for HIV and hepatitis C and many other viral polymerases. So it seems to be that organisms tend to change at a rate of about 1 change per genome copy. Now, a famous change that has been in the press a lot is this D614G substitution. As you can see, this is really a clade difference. So the sequences that arose in 2019 are all the aspartic acid D614 whereas in the spike protein, whereas the sequences that arose in 2020, and these initially seem to have started in Europe and then were transmitted around the world, these sequences all carry the glycine, the G version of this amino acid in the spike protein at position 614. And so the difference between these clades is in part that particular amino acid difference. Now, there are other differences that I'll show you, but when people talk about this polymorphism, you have to realize that this isn't arising in each person. This arose in one major event and has been preserved in the strains that have evolved subsequent to that. Now, our sequencing is depicted here, and I'm just showing you that in Maryland, there were this many positive tests in the state of Maryland with the unfilled bars, and then the positive tests at Johns Hopkins were the black filled bars. And so you can see that Heba's lab did more than half of the testing in the state of Maryland up until about the end of March, when other sites started doing more testing. So initially, we were really sampling most of the epidemic strains in Maryland. You can see that these CT values, which are a measure of the amount of virus present, that around 4 days after symptom onset was the peak in viral level in these NP swabs, and then that decayed over time. And you can see the age distribution here, where we had a lot of people in middle part of the age range, with very few people 60, 70, 80, 90 years old. And so it's worth noting that as we move forward, increasingly people are not going to be using CT value, but rather this unit called NDU, NAT detectable units per ml. And so this is going to get standardized, but for now we talk about CT values. And I'm just contextualizing this so you can understand where this is coming from. And so in this plot, we have one row per sequence. And so there are about 114 sequences here. So there are 30,000 columns across. And in red are polymorphisms relative to the Wuhan 1 strain. Gray and blue patches are areas of uncertainty. And so you can see that in most sites, for most sequences, we had some confidence, and that there are some clade. So this would be, for instance, the D614G change is this one here, which is shared by all the ones in this 20 clade, whereas the 19 clade, that polymorphism is not observed, but there are other changes in the genome that are linked. And so when we talk about these amino acid differences, realize that if we talk about a characteristic of a virus that has D614G, that virus also has P314L in ORF1b, and it also has F924F, a synonymous change, so a nucleotide change that doesn't change the amino acid at position 3037 in the genome. And so all of these changes are coupled in ways that make this complex to interpret. And any conclusion that a given amino acid change causes some difference in phenotype has to be understood in the context that there are probably other changes in that genome as well. And you may not be independently comparing that one change. So I won't belabor this too much. These figures are getting complex. They're all in the publication that's in Metacrive. But I'll just point out that one of the things we did was look at clinical parameters. So their disposition, where were they? Did they have a known COVID contact? Had they travel history? What was race and sex? Their symptoms, and we had a variety of symptoms coded, fever and shortness of breath, use of nonsteroidals, which was a hot topic early on, abnormal chest x-ray, comorbidities, which are listed here, and then outcomes, hospital admission, supplemental oxygen, ICU admission, ventilator administration. We originally had a row for death here, but there were so few that the folks who are honest brokers for privacy at Johns Hopkins felt that we could not include death as an outcome because the numbers were so small that re-identification risk was substantial. And so we had to remove death as an outcome from this analysis, where one column was one person, because of concerns about risk of re-identification. So all of our metadata that came out of this publication had to be cleared through a process of de-identification and approval, and our ability to describe some of the outcomes was limited because of issues of re-identification. So there are a number of inferences here. One, to say that if you look at any given zip code, so this is 3-digit zip code region, all the different clades were present in most of the zip codes that we looked at. So there's a lot of diversity. This wasn't simply founder effect with growth locally, but rather multiple introductions to every one of these, because the virus cannot change between clades in evolution. It's once the clade is established, they stay faithful to that clade during evolution. The other point here is this is a histogram violin plot looking at the Hopkins, Maryland sequences. And the point of this blue violin is to point out that the median distance, as well as the bimodal distribution of this distance, where moving to the right means greater diversity, the sequences from Maryland were as diverse as the sequences obtained worldwide, whether you looked at 1,000 or 3,000 genomes that were chosen multiple times in a Monte Carlo-like simulation. What you see is the distribution of diversity was great in Maryland. So we had an opportunity at least to link outcomes with genomics. What I'll say in short is that the analysis with just our 100 or so sequences was not powered sufficiently to find an association between clinical outcomes that we looked at and the genomics. It's possible that a larger data set would give a better answer, and I'll be able to touch on that in a moment. So this is just depicting variability in globally and in Maryland. You can see that the same sites that were variable in Maryland were variable globally, so it's representative. And so now I'm going to just show you some of these tools. And so if I click on GISAID, and I'm going to have to unshare for a moment, and then I'll share my browser so you can see these things. So this is the GISAID site, and you'd be able to log in and examine the sequences in GISAID. And as I said, there are more than 85,000 of those now. Now, if we go to next strain, this is, again, I'm not logged in. This is just a freely available site. And you can look at the resources for SARS-CoV-2. And this is the latest global analysis. And so as this loads, it's loading a sample, a representative sample of the GISAID database selected to be representative of frequencies around the world. And so here you can see the same sort of tree that I was showing before. And I'm going to change the dimensions of this window so it displays a little better for you, I hope. So it'll redraw. I'll just there we go. Okay. So what you'll see then is this rectangular cladogram. I'm going to change some of the options just so you see how interactive this is. So this is the novel coronavirus globally, and we can color by country. And if we color by country, then we see the same kind of color scale that we saw before, where it's ranging from purple to reds. And you can see now it's complex with many countries represented. I'm going to switch to showing you this coloring by genotype, so I can show you that if you color using the spike protein and position 614, then we can see that this change from aspartic acid to glycine that's been so prominently discussed, and I'll discuss in a moment, is really a split between the 2019 and 2020 clades. Now, it's worth saying that these 2019 clade sequences are still appearing, so this is still spreading, and you can see this group here is still spreading. And if you mouse over this, you can see that this is a strain from Singapore from September 3rd. So these sequences are still traveling around, and you get some detail when you mouse over these, and you can click on these and then learn more. We can also see this same tree as a radial tree or an unrooted tree. And as I mentioned before, we can also switch to the clock presentation. And here we see the regression of time on the horizontal axis against mutations on the vertical axis. And you can see that the rate estimate is still around 24 substitutions per year. And you can also see how the 2019 sequences are primarily found early on, and 2020 clade, this glycine has now replaced that largely, although they're still the earlier type appearing. And all of these are new isolates where you see green in this background. Now, if we go to, if we scroll down, then we see the global map. And if we hit play, then we'll see, and I don't know whether this animation will project well, but what we see here is the date and the time. And so what you see over time is that the virus is evolving and shifting from its initial origins in Asia through Europe to North America, Africa, and other places. And you can see that the yellow, the glycine version of the virus has replaced largely the earlier greenish aspartic acid version. And so, so that movie plays out if you just ask it, if you just request it. Now, the other thing you can do is click on this diagram of the genome, and you can choose any polymorphism you like. So here's the D614G mutation, but you could also, for instance, look at this 314 change in ORF1b. If we click on that, the tree will recolor. So now we have a new tree. We can go back to the rectangular cladogram, and we can see that this 314 site in ORF1b is also changing, like the site, the 614 site in Spike. And so these are co-varying, indicating that when we look at a difference between the D614G strains, we're really looking at a combination of changes that are linked. And just so you can explore this on your own, you can see you can filter by country. So you can click on any of these text descriptions, administrative divisions, the locations, authors. And so if you scroll down here, you could find Thielen, our first author, and you'd be able to highlight the sequences just from Johns Hopkins, for instance. So there are many tools here, and I'll leave it to you to explore this further, but I just wanted to highlight that for you.
We're getting a great comment in, which is, this is amazing. Dr. Ray is answering all of my questions as they occur. I'll still need to rewatch this at 0.75 speed later on to gather all the info. But could you please speak to this significant change with the yellow and the glycine? And for a non-clinician, what does that really mean?
I will, that's my next thing. So the question is anticipating where I'm headed. So that's great.
Perfect. Great.
So I'm going to reshare the slides. And so we're back to this set of links, and I'll highlight virological.org, which is another source of unvarnished discussion of the evolutionary considerations that we've been talking about. So there's a lot of people who have commented on the evolution starting from January and February. And so many of the things that have been discussed, including this spike protein polymorphism at 614, have been discussed there in some detail, and the origins of this virus. So if you want to see a debate of these things, that's all there. So there are some inferences that I have on this slide that I'll let you read if you're interested at leisure. I don't want to spend too much time on that because I want to touch on some of the other metadata issues that I presage. So here's a map just of the spike protein. And just to say that this protein forms a big part of the virion that we talked about. This is the transmembrane region. So this is the anchor of this protein in the membrane of the virus. So this part would be in the cytoplasm, and this part will be in the cytoplasm of the cell when it's being made. And this part would be outside the virion. And there are two major domains here. There's the S1 and S2. There's a cleavage here that occurs, and then there's a second cleavage that occurs here. This is the receptor binding domain. So this is the part that binds to the ACE2 receptor on human cells and is an essential step in the entry of this virus into our cells. This change that we've been talking about, the D614G, aspartic acid to glycine, occurs here, which is not in the receptor binding domain. This doesn't directly affect the binding of the spike protein to the ACE2 protein. What it appears to do, as I'll talk about in a moment, is stabilize this S1 domain so that it stays attached to the virion even after this cleavage occurs. and that appears to affect infectivity of the virus. And so here you see the temporal shift from this earlier D614 version to the G614 version in spike. And you can see this is frequency globally, that the 100% of the strains in early January were the aspartic acid version, the D version, and then that really flipped to the G version of this. And so one might wonder what caused this flip, and that's been an area of some great interest. So prior to March 1st, this is what the map looked like, with mostly the D version, the glycine, or the aspartic acid. And then in the March 21 to 30 period of time, the flip was occurring to the G version of this sequence, suggesting that something had occurred, but interestingly, it occurred everywhere that the virus was present. What did not happen is just the change at the one specific site, because as I showed you in ORF1b, there's that 314 change that's coupled with this one. And so these, what did not happen was that strains that lacked the glycine changed to glycine. What happened was that both strains were present in many regions, and the glycine version replaced or became the dominant strain in all of those places. And so the question is, what was the reason for that? And so this is depicting in Asia, Europe, North America, Oceania, South America, Africa. In all these places, early strains when present were the aspartic acid version, later strains were the glycine version. And so this happened globally. What's thought to be true is that this change increases the infectivity of the virus by stabilizing that S1 domain. But this is perhaps one of the more interesting things that I've run across recently. This is a report from July from Drew Weissman's group at Penn, where they demonstrated that vaccinated mice, non-human primates, and humans who have recovered from infection, or humans who've received the mRNA vaccine that includes the spike protein, that this plasma from all of these organisms more effectively neutralizes the glycine version, the one that evolved, than the original aspartic acid version. And so as we think about this, we might wonder, well, why would the virus evolve toward having something that's more easily neutralized? So there's another study that also did sequencing. This was in Houston, at Houston Methodist, and they also found replacement. They had an early wave of COVID in Houston that was that included this aspartic acid version, and then the glycine supervened. And so they observed the same kind of phenomenon. And they also found that people who had the aspartic acid version of the virus had lower viral levels in their NP swabs than people who had the glycine version. So in multiple studies, it's been found that the glycine version appears to be more infectious in the early parts of the epidemic. So in this shift, it appears to be attributable to an increase in infectivity of the glycine version, which may explain why the virus changed from aspartic, or the dominant strain shifted from the aspartic acid version to the glycine version. However, on the previous slide, I showed you that this glycine version, the red one, is more easily neutralized. And I think it's an important insight into this whole epidemic that no humans were immune to this virus before it entered the human population. There's some cross-reactivity with existing coronaviruses, and so while there's some degree of immunity that may help protect people, the reality is that nobody had antibodies to the spike protein that were neutralizing when this virus first entered the human population. And because people tend to transmit the virus within a week of getting infected, there was no time for them to develop an antibody response. And so the virus is continually being transmitted to people who are not immune. And so the virus has never really been pressured by neutralizing antibodies that have been generated by humans to this virus. The implication here is that when more and more humans are immune to SARS-CoV-2 because of prior infection or because of vaccination, once we have vaccines, we may see a shift back from this glycine version back to the aspartic acid version, because as Drew Weissman's group showed, this version is more resistant to neutralization than the glycine version. So one way to put the story together is that in its previous bat host, the aspartic acid version was more common because those bats had some immunity to spike. But going into a naive population, the human population, the virus has been running rampant with no controls immunologically. And so the glycine version, which is more infectious, is spreading. But once we have some immunity, we'll see a shift back. Now, to answer the question about the linkage between metadata and these genetic variants, here's an example where the Department of Defense has the ARMOR-D program, where they link repositories and dictionary tables and isolate data from people, including bacterial isolates in this case, where they generate results that include a variety of characteristics, including antimicrobial resistance and bacterial strain data. And of course, there are many bacterial strains in this model. And what they've done is encrypted the data about the people. So in this schema, they're really interested in antimicrobial resistance, and they spend less time looking at the human metadata. And so the metadata have been put off in an encrypted vault that's not accessed as readily as the bacterial resistance data and phenotyping. So the question is, how should we model a system where we do similar work, but in the case of the coronavirus, we think that the viral sequences could have an important impact on pathogenesis and our understanding of spread. And so we might need to have more of this human data co-located with the viral data. And the solution for this is not yet obvious, which was my motivation for talking to you all about this. So one question that was raised during our discussion with the National Academies is, should restrictions on metadata sharing be relaxed in a pandemic? Or should we make our data sharing infrastructure more robust generally? Because as it was, the GISAID database has some data, but it's been jittered to de-identify it. And it has very little clinical metadata associated with it. And so we have all this genomic data, 85,000 genomes with very little clinical metadata attached. Then we have N3C, which Chris Shute has talked to you about, and you'll hear more about, which is a major national collaborative of COVID data, which has huge amounts of clinical data, but does not yet have much viral metadata. And perhaps that's the best place to link these. However, that's in an enclave that people can't download. And so the tools for analyzing the data would have to be added to that analytical infrastructure, or we'd have to find other ways to link inferences between the genomics and the metadata. And then there's also the possibility that in a pandemic setting, public health authorities like the CDC, which does not have restrictions on the receipt of protected health information, could aggregate population-based sampling, which of course they can do where a given health system cannot, the data aggregation, and support downstream research efforts that depend on the annotated viral genomics. I don't think there's a coherent solution to this right now. And so I'm gonna flip to my last slide, which is just some questions for us to consider going forward. But I think the hard question is, what's the right way to link the clinical metadata, and where would we do this with viral genetic data when what we really need is population-based sampling of both? And I don't know the right answer to that. So I'm gonna open it up. And if Ash, tell me whether you think I should stop sharing, or how do you want to do the Q&A going forward?
Yeah, that's great, Stu. I think these hot topics are ripe for questions, and I know that we've been getting a few. I'll throw it to Paul right now to pose one of the questions that came through. And yeah, if you want to stop sharing, that's great.
Well, I leave that screen up because I think this really raises a lot. I think you posed it really well. It was really a tragedy when you couldn't get the sample size. You've done all this investigative work, sequencing, and we have some outcomes data, but because they're being stripped, the outcomes data is in medical records, and it follows a different path than the sequencing data. And this is a classic story of the innovation happens when you match the two up. This is the peanut butter and jelly story, you know, and I really like that you draw the linkage to last month's grand rounds for how do we start sharing metadata. But right now, that's just the outcomes data. That's not these key forensic sequencing data. And you're right, trying a link between the CLADE and the WHO severity scale for how sick the patient gets. It's really it really strikes me, you know, that we don't have the infrastructure to answer that question, where we can scale it across multiple institutions. And so it seems like you know there's a great infrastructure for how you create linkages and you submit it to GIS aid, and but then that does not get tied to the metadata for the outcomes. And so it is interesting how you're approaching this, and I'm hoping. I'm hoping N3C could be the place that we could provide that linkage, but it is interesting that there's no, you're right, there's no obvious solution to this problem outside of a single institution.
And you notice the next strain visualizations are really nice. They're robust, but they don't include much clinical data because of the limitations on sharing. And so if you could just populate that database, there are armies of programmers who work on NextStrain who would be happy to make visualizations. And often those people are starved of clinical metadata because they're not in they're not in a covered entity. So they don't have access to these kinds of data. And there was a lot of frustration actually initially when people said, you know, gosh, why can't you give us more metadata? And the answer was HIPAA. You know, that we really have to preserve patient privacy. There's a good reason for preserving privacy in an epidemic setting, but boy, it's tough. Now, you can all imagine that if we shared the data freely and we added key characteristics like a person's diagnoses and a lot of other metadata to the sequences and deposited those in real time for all infections, there might have been a lot of interest in somebody living near 1600 Pennsylvania Avenue who might have a bunch of diagnoses attached to their sequence, and people would do their best to figure out where that person was and how many diagnoses they can glean from that.
Yeah, it's the Massachusetts governor story all over again, where they
yes,
were able to
exactly.
re-identify him through his clinical trial profile.
Yes.
I do think, though, that this is actually a good fit for OMOP within an institution.
Yes.
There's strong ontologies for the sequencing data for representing it, and so I do think this would be where we could actually pass the queries amongst multiple institutions and do acyclic graphing type of federated learning without actually having to aggregate the data. So it seems like this would be a great fit for bringing up, and just for our students, this is the future of innovation, bringing two different data sources together that haven't traditionally really worked well together, whether it's imaging, sequencing, physiological monitoring, or EMR outcomes data. There's really immense innovation to be had when we can begin to really combine them effectively at scale to make new discoveries.
Another question that's coming through is regarding the National Academy of Medicine publication and the link that you included. And the question is, what has been some of the initial feedback that has been received as a result of that publication?
As far as I can tell, there's been a deafening silence. It's, you know, we tried to be provocative. If you know anything about that process, it's a tricky process because you try to make recommendations, and the National Academies is careful because in general, they cannot recommend appropriations to Congress. So they can make recommendations for use of resources and that sort of thing, but things that would require new expenses are complicated to recommend. So we could talk about principles and best practices, but it was hard to make concrete recommendations that would result in a recommendation for appropriation in Congress. So I think we felt that N3C is an amazing opportunity for linking data together, and maybe we should do more and more viral genetic data there because that is a place where we can do that. The tough part is that's generally covered entities or enterprises that have access, and so you're not going to get a true population-based sample. Right. You'll have a sampling of the institutions that happen to participate in N3C, which is a different thing.
Yeah. Yeah, the second route would be like NIH all of us, right? Would that be,
Yes.
that'd be a little bit closer to a population sample where, but that's all tied to volunteers and people willingly submitting their records. So it has its own type of bias.
And my guess is right that there's going to be a socioeconomic bias in the all of us inclusion. Although there has been some deliberate effort
Yes.
to have racial diversity and ethnic diversity, I think socioeconomically is probably not fully representative. The CDC and its inputs probably are much more population-based because they have all the public health clinics and other places that are a little less split that way. And they have access to all of the specimens from all of the entities as well. So I think that would be the most likely input pipe for specimens and potentially metadata because they can collect it. The challenge is they're underfunded. And so it's hard for the public health authorities to receive robust data because they don't have means to import them. And they're amazingly, they often work by fax machine. So a lot of what the public health authorities do is by fax.
Believe it or not, we're at the top of the hour. I think the two posts from Dr. Chris Shute and Dr. Harold Lehman are relevant to wrap up with. Chris Shute, who was our Grand Round speaker last month, writes that N3C has invested considerable efforts in data linkage and hashing, and some N3C contributors are entire states. And Dr. Harold Lehman points out that N3C has a domain team, social determinants of health, working exactly on the population issues of the N3C selected sample. So Stu, thanks so much for taking your time to present at our second Informatics Grand Rounds. This really was a fantastic discussion. If folks have any follow-up questions at all, please feel free to email at us at jhinformatics@jhu.edu. Thank you all for joining us.
Yeah, thank you.
Thanks so
Great.
much.